Okay, I'm sorry again for the abrupt interruption to the last video. Okay, so we are discussing N-linked glycosylation, which is a process which occurs in the endoplasmic reticulum by which polysaccharide sugar units are added on to proteins which are in the membrane of the endoplasmic reticulum, and this process is known as N-linked glycosylation. Okay, so we now want to see how you build the structure which is going to be linked on, because it's a very specific structure that is linked on to the proteins, and we now want to see how that structure is built. So we've discussed this molecule dolichol, which is the molecule in the membrane of the endoplasmic reticulum on which we're going to build this sugar structure that we're going to add onto proteins. Okay, so far we've got dolichol phosphate, and the first sugar we're going to add on to that dolichol phosphate is going to be N-acetyl glucosamine. So I want to now show you the structure of glucosamine and then show you the structure of N-acetyl glucosamine. But in order to do that, we firstly need to have a reminder of the structure of glucose. So I'll draw this structure in skeletal form. Okay, so where I don't put the atoms, that denotes that it should be a carbon. So here's an oxygen here. So glucose is based on a six-membered ring. Well, the form of glucose that we're going to look at, glucose actually can exist not in a ring structure at all, and it can exist also in five-membered ring structures. But the type of glucose we're going to look at, which specifically is known as beta-D glucopyranose, which is the specific isomer of glucose that we're going to look at, it exists as a six-membered ring. Okay, and then what you have is you have a methyl group which is coming out of the page at you, basically. So if you imagine this ring in the page, then we're going to have this next group coming out of the page at us. Okay, so I'll denote this with this sort of line that gets thicker towards its end, and that shows us that it's coming out of the page. Okay, so this is CH2OH. So it's basically a carbon with two hydrogens linked to it, and then a hydroxyl group linked to it as well. Okay, and of course the other group that's off that carbon, that fifth carbon of the glucose ring as it's called, so this would be the first, this would be the second, this would be the third, the fourth, the fifth, the sixth. The other group that's coming off this fifth carbon would just be a hydrogen, so we won't denote that. Okay, then you have another hydroxyl group coming off here, but this time it's going away into the page, so we'll draw that like so. So a dashed line will denote that it's going into the page. Okay, now this next group down here, this hydroxyl group will be coming out of the page, so again I'll draw it with this bond that's getting thicker towards the end. So this is coming out of the page at us, and then another hydroxyl group off this second carbon will be going into the page away from us. And finally, the hydroxyl group coming off this first carbon, that will be coming out of the page at us, like so. Okay, right. So, this is glucose, but the specific isomer of glucose that it's called, if you want the specific name, it's known as beta-D, and then glucopyranose. Glucopyranose. Okay, so the pyranose denotes that it is a six-membered ring here. The beta denotes the fact that the hydroxyl groups on this first and fourth carbon are in opposite orientations, basically. So if this was alpha-D glucopyranose, then the hydroxyl group here will be going back into the page as well, whereas in beta-D glucopyranose, you have them basically, this one is going into the page and this one's coming out of the page, so in opposite orientations. Okay, and this isomer of glucose is what we're going to base glucosamine on. Okay, so glucosamine then. Glucosamine. So basically it's this exact same structure as glucose, so let's draw it out again, this six-membered ring on which glucose is based. Here's the oxygen up here, and then the other five members of this ring, 1, 2, 3, 4, 5, are all going to be carbons, and then here's our sixth carbon up here, and that's coming out of the page again, so we'll denote it like so, to keep our, because it is very, very important. You might be wondering, why am I faffing so much about whether they're coming in or out of the page? It is important. When we look at mannose, you will look at mannose's structure and you will say that's the same as glucose, and I will say no, it's not. The orientation of these hydroxyl groups coming in and out of the page is different in mannose, and that's what separates it from glucose, and we'll see that later because mannose is going to be another one of these constituents which we're going to use to make our sugar group that we're going to add onto proteins. Okay, so up here you have this CH2OH group here. And then coming out here, you have this hydroxyl group coming off the first carbon. And because we based it on beta glucose, this is basically coming out of the page as well. So here we are. Then we've got a hydroxyl group off this second carbon, which is going into the page. A hydroxyl group of the third carbon, which is coming out of the page. Oh, whoops. Sorry, I'm just getting a little carried away here. I've just basically drawn the structure of glucose out again. This is going to change in glucosamine. So get rid of that hydroxyl group and instead put an amino group on there. That is how you change glucosamine from glucose. Okay, and then you have a hydroxyl group going into the board there. Okay, so this is what makes it glucosamine, because you've got an amine group there instead of the hydroxyl group. Now, if we want to make N-acetyl glucosamine, what this means is we're going to esterify an acetic acid group to the glucosamine monosaccharide. Okay, so acetic acid then, the structure of acetic acid is as follows. Acetic acid is the old name for ethanoic acid, so let me show you its structure. Ethanoic acid means that you have two carbons, it's the 2-carbon carboxylic acid. But, as I say, biochemists' name for this is acetic acid. Acetic acid was the old chemist's name, but chemists have renamed it ethanoic acid, which is more sensible because it follows a very nice naming protocol. But the old name for this was acetic acid, and that's still a very pervasive name that you'll see used in biochemistry. Okay, so basically to create N-acetyl glucosamine, you're going to link an acetyl group of the glucosamine, and it's going to link in an amide bond. I'm sorry, I said ester before. It's going to link in an amide bond. So what's going to happen is if we draw this amine group out in full, it has these two hydrogens coming off this nitrogen. What's going to happen is you're going to take that hydrogen, one of these hydrogens off this nitrogen, you're going to take the hydroxyl group off the carboxylic acid group. So let me get some colour on here. Okay, so you're going to take this hydrogen and this hydroxyl group off of these chemical molecules. They're going to go together to make water. That's why it's called a condensation reaction, because you're going to produce water. And then you're going to link this carbon here, okay, to this nitrogen here. So that then will give you N-acetyl glucosamine. So let's draw its structure out one more time, just to have a bit of a revision. So again, this 6-carbon ring here, okay, 6-membered ring with only 5 carbons in, okay, and then we have our 6th carbon coming off up here, which has this CH2OH domain here, okay, and I'll draw that, like so, with it coming out of the page. Okay, then you have this hydroxyl group coming out here, because remember it was based on beta D-glucopyranose rather than alpha D-glucopyranose. In alpha D-glucopyranose, the hydroxyl group would have been going into the page. And then going into the page, we then have this amino group, which has become linked by this amide group to our acetic acid group here. Okay, and then you have a hydroxyl group coming off carbon 3 here. Okay, and that's coming out of the page at us. And then finally, off carbon 4 over here, you have a hydroxyl group going into the page. So this is N-acetyl glucosamine. This now is the structure of N-acetyl glucosamine. And because N-acetyl glucosamine is a bit of a mouthful, often people will just abbreviate N-acetyl glucosamine to NAG, or NAG for short. So this is often abbreviated to NAG. Right, okay, so how are we going to link N-acetyl glucosamine to our dolicol phosphate molecule? Well, basically what we're going to do is we're going to bind our N-acetyl glucosamine to a UDP molecule, okay? And UDP, we won't go through the entire structure of UDP. Instead, what we'll do is we'll draw it as a little bit of a cartoon. So, we'll denote UDP as so. UDP stands for uracil diphosphate, okay? And it's going to, well, actually the way you're going to bring it on is you're going to bring on UTP. The way you're going to bind it initially to the NAG is you're going to have UTP. So I'll draw UTP initially. So uridine triphosphate is what it stands for, and basically it consists of uridine nucleoside. So I'll just explain what a nucleoside is. Right, so here's the structure of UTP. Now, uridine is what is known as a nucleoside. Okay? Now you might be wondering what's the difference between a nucleoside and a nucleotide. Nucleotide is the one that generally people have heard of. Nucleoside is the one that less people have heard of. Nucleoside is the name for the organic base along with the ribose sugar here. So it's this ribose sugar, which is this 5-membered ring along with the 5th carbon up here, because remember this first member is just an oxygen. And the organic base here. So the organic base in uridine is uracil. So uracil is the organic base. That's this bit here. Okay, so uracil is this bit here, the organic base. Uridine is the name for uracil bound to ribose, and it's an example of a nucleoside, which is an organic base bound to a ribose sugar. Now, what is a nucleotide? A nucleotide is when you take a nucleoside and you add on phosphate groups, basically. So the whole thing is then a nucleotide, and it's one with 3 phosphate groups on. Okay, so let me colour in these phosphate groups red. Okay, so that's why this entire structure is now known as uridine triphosphate, because you've got uridine, the nucleoside here, and then it's bound to 3 phosphate groups. So that's what the origin of its name is, uridine triphosphate, UTP. Now, basically what we're going to do is we're going to link this gamma phosphate, as it's called. So just to label these phosphates up, this terminal one here is known as the gamma phosphate because it's the 3rd one. So the Greek alphabet goes alpha, beta, gamma, and gamma is the 3rd letter. So this is the 3rd phosphate group. This would therefore be the alpha phosphate, and this would be the beta phosphate. So basically what we're going to do is we're going to take this gamma phosphate off, and we're going to link the beta phosphate then to this hydroxyl group here, which is how we're going to get uridine diphosphate, UDP, linked to our N-acetyl glucosamine molecule. Okay, so let me draw that reaction out. You're going to chop off this gamma phosphate here. You're then going to perform a phosphate esterification here. So you're going to link this phosphate group to this alcohol group here. Okay, so let me get some pens to denote that. It will be a condensation reaction, basically, and it will be catalyzed. The energy needed to produce that reaction will be given by the breaking off of the gamma phosphate, basically, because as we know, when we hydrolyze ATP to ADP in inorganic phosphate, it releases energy. Well, sorry, this is UTP, but UTP has the same principle as ATP. When you hydrolyze the final phosphate, it gives out energy. ATP is the more universal energy currency, but UTP is capable of doing the same thing. So, after all that discussion, what we have created is a molecule of N-acetyl glucosamine. So we'll have NAG here, which has its first carbon. The first carbon of that ring is now linked to UDP, uridine diphosphate. Okay, uridine diphosphate. Right, so that's what we've got so far. Right, so what we're now going to do is we're going to link this on to our structure that was in the membrane, which we were building off, a dolicol. That's what I'm looking for. Couldn't think of the word for a moment. Dolicol. Right, so we're going to link it to dolicol in the ER membrane. Okay, right, so if I draw the ER membrane here, okay, and we have dolichol here in turquoise, and dolichol had already had a phosphate group linked to it here. Okay, now if you remember, the way we linked N-acetylglucosamine, NAG here, to our UDP is that we linked it to that beta phosphate, basically. So, if we draw this in a bit more detail, what you've got is these two phosphate groups here, and then you've got the ribose sugar, like so. Okay, and then the organic base off here. And that's what we've actually got attached to our N-acetylglucosamine. Now, what we are going to do when we attach this N-acetylglucosamine to our dolichol over here, so this is our dolichol sitting in the membrane of the ER, and at the moment it's facing the cytoplasm, so this phosphate of the dolichol is facing the cytoplasm, and the other end, where you've just got this isoprenoid group, is facing the ER lumen. Okay, right, so what we're going to do basically is we're going to chop off UTP. So we're going to chop off, we're going to chop off UMP, we're going to chop off uridine monophosphate. So we're going to chop off the uracil organic base with the ribose sugar and this alpha phosphate. And then what we're going to link is we're going to take the N-acetylglucosamine with this phosphate group and bind it to this other phosphate group on dolichol. So what we're overall going to end up with is, if this is dolichol here, with its phosphate group here, then we're going to end up with it linked to another phosphate group, and then we're going to have N-acetylglucosamine bound there. Okay, so that now is the structure of what we have. Well, that's now so far what we have created. So we've managed now to stick on our first sugar onto dolichol. And then what we're going to continue is we're going to add more sugars onto the N-acetylglucosamine, and we're going to form glycosidic bonds in the way that you would usually between monosaccharides. Okay. Right, so let me just remind you of the structure of N-acetylglucosamine so that I can show you what we're going to do next, because we're going to link more N-acetylglucosamine molecules on the end of this to create a whole sort of chain of sugars basically bound together by glycosidic links. Okay, right, so let me remind you of the structure of N-acetylglucosamine then. So the structure went like this. Oh, shall I draw it? Yes, I'll draw it skeletally. We'll use that as the sixth carbon. So here's the six-membered ring with the oxygen up here, like so. Okay, and then we had a hydroxyl group coming out of the page here at you. So this was a hydroxyl group coming out of the page at you, and then going into the page away from you, you then have this amino group, which have been linked by an amide bond, remember, to our acetic acid. So here is our N-acetyl group, basically. Then, off this third carbon, over here, let's draw that there, we have this hydroxyl group there, okay, and then going into the page off this fourth carbon, you then have a hydroxyl group, okay. Now coming out of the page off this fifth carbon, you have your sixth carbon of the N-acetylglucosamine ring. Okay, so here's our sixth carbon, CH2OH, up there. Okay, so that's the structure of N-acetylglucosamine. Now, basically, this N-acetylglucosamine here has these two phosphate groups are linked off this first, this hydroxyl group off the first carbon. Now, it still has its fourth carbon available. So what we're going to do basically, it has its fourth carbon hydroxyl group still available. So what we're going to do is basically bring in another N-acetylglucosamine and link the hydroxyl group that is off N-acetylglucosamine's fourth carbon here to the hydroxyl group of another N-acetylglucosamine. So we're going to bring in another one and form what's known as a glycosidic link, which is basically when you perform a condensation reaction between these two hydroxyl groups. Okay, so we're going to glycosidically link more N-acetylglucosamine molecules to our structure, basically. Okay, so we're going to add on another one here. N-acetyl glucosamine. And it's going to be linked by this glycosidic link. So the hydroxyl group of the first carbon of this one is going to form a condensation, glycosidic link with that hydroxyl group of the fourth carbon of this N-acetyl glucosamine. And that is going to result in the two being linked together. Okay, and we'll continue this discussion in the next video.